In 2022, a dish of living neurons learned to play Pong. Not a simulation of neurons. Not a neural network in software. Roughly 800,000 actual brain cells, some human, some mouse, grown on a chip studded with electrodes, wired into a video game from 1972. Within 5 minutes, they were hitting the ball back. That experiment is the reason you keep seeing the phrase brain organoid in headlines. And it is the reason most of what you have read about brain organoids is subtly wrong, including, as we will get to, the assumption that the Pong experiment used one. There is now a company selling a computer that runs on human neurons. It costs about $35,000. You can rent time on one over the internet today. So let us be precise about what these things are, what they can actually do, and where the real line sits. Start with the thing itself. An organoid is a small, simplified, three-dimensional clump of tissue grown from stem cells. You take stem cells, cells that have not decided what to become, and you give them a specific sequence of chemical cues. Then you mostly get out of the way. The cells self-organize. They sort themselves into layers, form cavities, and differentiate into the cell types of a particular organ. Nobody assembles them. They build themselves, following instructions that are already in the genome. This works for gut, liver, kidney, retina, and it works for brain tissue. A brain organoid is typically somewhere between a poppy seed and a pea, a few millimeters across, containing hundreds of thousands to a few million cells. Under a microscope, it looks like what it is, a pale, roughly spherical blob. It contains real human neurons, firing real electrical signals, forming real synapses with each other. That last sentence is completely true, and it is the source of nearly every misunderstanding that follows. Here is where almost all coverage of this field goes wrong, and it is worth 30 seconds of your attention because everything downstream depends on it. The Pong experiment did not use a brain organoid. It used a two-dimensional sheet of dissociated neurons, cells grown flat across an electrode array, like moss on a stone. They were connected to each other, and they were alive, but they had no three-dimensional structure. No self-organization. That system was called DishBrain, and the company behind it is Cortical Labs in Melbourne. A brain organoid is the other thing, the self-assembled three-dimensional ball from the last section. Both are living human neural tissue. Both can be wired to electrodes. But they are not the same object, and the difference matters, because the flat sheet is easier to interface with, while the three-dimensional organoid is the one that actually resembles developing brain tissue. Keep both in your head. Different groups are pushing on each. So how do you teach a dish of cells to play a game? The array does two things at once. It can record electrical activity from the neurons, and it can stimulate them. The researchers mapped a region of the array to ball position, stimulating cells in a spot that corresponded to where the ball was. Activity from another region moved the paddle. Then the feedback. When the paddle hit the ball, the neurons received a predictable, regular stimulus. When they missed, they got an unpredictable burst of noise. That is the whole training signal. Predictability as reward, randomness as punishment. The idea underneath it is that neural tissue reorganizes to minimize surprise about its environment. Give a network a world it can predict by behaving a certain way, and it will drift toward behaving that way. Within about 5 minutes of gameplay, rally lengths increased. Now, and this matters, a number of neuroscientists pushed back hard on describing this as learning, and harder still on intelligence. What was demonstrated is that neural tissue adapts its firing in response to structured electrical feedback. Whether that word is learning is a genuine, unsettled argument. Here is the part that turns this from a curiosity into an industry. In March 2025, Cortical Labs launched a product called CL1. It is a biological computer, living human neurons cultured on a planar electrode array packaged with the life support system that keeps them alive, temperature regulation, gas mixing, nutrient circulation, filtration. The array has 59 electrodes. The neurons stay viable for up to about 6 months, and the unit costs somewhere in the region of $35,000. Think about what that means practically. Biological neural tissue stopped being something you need a university wet lab to work with and became something you can order. The pitch is not that this outperforms a graphics card at matrix multiplication. It does not, and nobody claims it does. The pitch is efficiency and adaptability. And here the numbers deserve care because they get exaggerated constantly. A CL1 unit draws about 30 watts. A full rack of 30 units draws somewhere around 900. You will see it claimed that this uses less power than a pocket calculator. It does not. 30 watts is a dim light bulb, and a calculator runs on thousandths of that. The real comparison is still interesting. A single high-end AI accelerator in a data center pulls around 700 watts on its own. So an entire rack of biological units sits in roughly the same power envelope as one or two of those chips. That is a genuine efficiency story. It is not the thousand-fold miracle the headlines imply, and whether it becomes a real computing platform or an expensive dead end is still open. But the hardware exists, it has a price, and it ships. And you do not even need to buy one. Cortical Labs offers remote access to their systems over the internet. The term they use is Wetware as a service. You send your experiment to living neurons in a facility somewhere else, and the results come back. A Swiss group called Final Spark runs something similar, and theirs uses actual brain organoids, the three-dimensional kind, rather than flat cultures. Multiple organoids, each on its own small electrode array, accessible remotely by research teams around the world. So the field has quietly reached a strange milestone. Living human neural tissue is now a cloud resource. There is a queue. There is an API. The organoids themselves have a lifespan measured in months, and they have to be continuously fed and maintained. Something in a building on another continent is being kept alive so that someone can run a job on it. Sit with that for a second, because it is about to matter. Meanwhile, a separate line of work has been going after something different, not computation, but repair. In May 2026, a team at Cambridge published a system they call a corticospinal connectoid. They grew brain tissue and spinal cord tissue as separate organoids, positioned them near each other, and waited. Nerve fibers from the brain organoid grew across the gap. They reached the spinal cord tissue and formed a working circuit, and when the researchers added small clusters of muscle cells, signals traveling that circuit made the muscle contract. A lab-grown human brain-to-muscle pathway, firing. They kept it alive for more than a year, which let them find something clinically significant. Up to roughly day 150 of culture, corresponding developmentally to around the middle of pregnancy, damaged axons could regrow. After that window, regrowth collapsed. So they went looking for why, identified a gene network governing the shutdown, and then tested an existing hormone drug against it. Regrowth was substantially restored. That is the real promise here, and it has nothing to do with computing. Spinal cord injury is permanent because adult human axons do not regrow. This is a human system, outside a body, where that rule was bent. Now the part the headlines skip. A brain organoid is not a small brain. It is not conscious, and the scientific consensus on this is not close. Start with what is physically missing. It has no blood vessels. Real brain tissue is threaded with vasculature delivering oxygen to every cell. An organoid relies on diffusion, which is why it stops at a few millimeters. Beyond that, the center starves. The core of a large organoid is often literally dead tissue. It has no organized architecture. A human cortex has 6 distinct layers with precise wiring between regions. An organoid produces the right cell types in roughly the wrong arrangement. And most decisively, it has no input. No eyes, no ears, no skin, no body. Nothing to perceive, and nothing to act on. Whatever a brain is for, it is doing it in relation to a world, and an organoid has no world. What you have is human neurons firing in an arrangement that is not a brain, with nothing to think about and no means of thinking it. The honest position is that today's organoids are not sentient, that nobody has a reliable test for consciousness in tissue that cannot report anything, and that researchers in this field are uncomfortable about exactly that gap. So where does this actually leave us? Two things are true at once. Today's organoids are not conscious, and their trajectory is towards systems that are larger, longer-lived, better structured, and, most significantly, connected to inputs and outputs. Every limitation from the last section is a live research target. Vascularization is being worked on. So is layered structure. And the Cambridge result is precisely an organoid gaining an output, a muscle it can move. Nobody in this field thinks a dish is about to wake up. The concern is subtler and more awkward. We are steadily removing the exact features whose absence is our current reason for confidence, and we do not have a test that would tell us if we went too far. That is the genuinely interesting problem. Not a brain in a jar. A slow accumulation of capabilities, none of which is alarming on its own, with no agreed threshold and no alarm that sounds. The neurons played Pong in 5 minutes. Working out what we owe them is taking considerably longer.